Okay, first of all, thanks, Zhuzhou, for your kind introduction. So again, hello everyone. My name is Jiachen. Maybe I just hold it. I'm a second-year master's student in bioinformatics. I am happy to share my favorite project throughout my short research career with you today, which is ridiculously simple, but it answers an essential question of interest, which is how repeatable is phenotypic evolution? It's not. A lot of noise. Okay. So let's imagine a scenario where we evolve the same populations under the same environment. What is the outcome in the end? Well, there's a fundamental tension between two factors that may alter the outcome of evolution. On one hand, evolution involves several stochastic processes. Mutations occur randomly, first of all. Second, the fixation of the loss of the mutant alleles is also random. Third, the appearance and the fixation of a mutation affects future mutation. This is a phenomenon called as contingency. Well, the contingency itself is not random. The outcome of contingency is. On the other hand, there are also deterministic forces. Because we evolve the same population under the same environment, the organism face the same selective pressure. And also, we have the general prediction that fitness generally increases during evolution. So, because the relative contribution of these two factors largely depend on specific conditions, such as your starting genotypes, mutation rate, population size, and etc., the repeatability of evolution becomes an empirical question. So our thought experiment has been already famously proposed by biologist Stephen Jay Gould in his book Wonderful Life. He used the metaphor replace the life's tape. Oh, this is really annoying. This is clip it. Okay. Oh, this is too close. Is it better now? Can everybody hear me? Oh, sorry for that. So he used the metaphor of replay life's tape, which basically means that reverting everything on Earth to their original states and let them unfold again under the same condition. But later in his book, he regretted that we can never possibly perform such experiment. But now here comes the opportunities nowadays. We can truly replay the tape in lab by experimental evolution. So because a lot of folks are from EEB and you should be very familiar with experimental evolution, I will just quickly go through how experimental evolution is typically done. So let's say we started with this founder population. We constructed, in this case, 4 replicated ancestral populations from the founder population. We measure the phenotype of interest of these ancestral populations, and we allow them to evolve for X generation in an environment Y. So at the end of the experimental evolution, we have 4 evolved populations. Then we measure the phenotype of the evolved populations. Then we compare against the ancestral population to evaluate the selective pressure imposed by that environment Y. Because such replay experiment is so easy to carry out, yet it is very powerful. People have previously carried out, previously carried out some replay experiment. The most famous one, which you might already know, is E. coli long-term evolution experiment established by Richard Lansky at Michigan State University in 1988. So in brief, 12 replicated populations of E. coli were allowed to continuously evolve in glucose minimal medium since 1988, sorry. And this experiment is still ongoing today. And one of the studies based on this E. coli long-term evolution experiment found that among 12 populations, only one population evolved the ability to grow aerobically on citric when the population reaches 50,000 generations. So it seems that divergent phenotype can emerge, even though we started with the identical conditions. Well, this is not uncommon. For example, there's another early study published in Nature. They evolved some identical lines of green algae in elevated carbon dioxide. After the experimental evolution, they found, where is my pointer? Let me get my laser pointer. A lot of lag, sorry, I'll just point it out. So they found for some lines of green algae failed to evolve specific adaptation to elevated carbon dioxide. Some lines even evolved a syndrome involving high rates of photosynthesis and respiration combined with higher chlorophyll content and reduced cell size, while other lines didn't. There's another example published in Genome Research. So in this paper, the authors evolved 7 lines of E. coli K12 strain in glycerol and 7 lines in lactate. And they found that for the adaptive evolution on both lactate and glycerol showed convergence of growth phenotype at the endpoint of experimental evolution in 6 of the 7 evolved populations. So here, the growth phenotypes here refers to growth rates, substrate uptake rates, and oxygen uptake rate. So as you might notice, such observation of divergent phenotype emerging from replay experiment is not uncommon, as I just mentioned. But you might notice that the number of phenotypic traits considered per study is relatively small. Usually people just focus on a few organismal traits. Is there a better approach to investigate the repeatability of phenotypic evolution such that a large number of traits are considered simultaneously? So here we came up with a better approach, which is to use transcriptomic data because the expression level of every single gene can be regarded as a phenotypic trait. So in one sequencing run, I can obtain thousands or even hundreds of thousands of gene expression traits. I did some literature searches and I found 10 experimental evolution data sets that are suitable for my purpose. Among these consist of 1 prokaryote species, which is E. coli, and 5 eukaryotes. We have yeast, copal, fly, guppy, and morning glories, spanning 22 distinct environments. And among these studies, gene expression is measured at 2 time points. When the organism first entered a new environment and at the conclusion of the experimental evolution. So what I did was I compared the later time point with the first time point to assess that whether for a gene, its expression has significantly changed after the experimental evolution. Okay. I've been talking about repeatability. So here, let me give 3 formal definitions of evolutionary repeatability during gene expression evolution. So the first definition is the sharing of differentially expressed genes, DEGs, among replicate populations evolving in the same environment. The second definition is the sharing of DEGs with the same direction among replicate populations. And the third one, the most stringent criteria, is the sharing of DEGs with the same direction and magnitude among replicate populations evolving in the same environment. Just want to better visually illustrate what I mean by 3 definitions of repeatability. So let's consider this simple example. So let's look at gene A first. For gene A, after the experimental evolution, its expression has significantly changed in these 2 replicates, but they change in different directions. For this gene A, I satisfy my first definition of repeatability. For gene B, so after experimental evolution, the expression has increased in both replicates. So I say for gene B, definition 1 and 2 are satisfied. For gene C, and also works if the expression significantly decreases in both replicates. That is also true. For gene C, after the evolution, its expression has significantly increased by the same magnitude. So I say for this gene, it satisfied all 3 definitions of repeatability repeatability simultaneously. Okay, so after we know how repeatable a gene is, we need a control, so a baseline to compare with. So I need to estimate the repeatability just by chance. So how do I do that? So again, let me put out this example data frame. What I did to estimate random expectation is I shuffle the columns independently. So by just shuffling, all the biological associations are broken in this case. And I did the shuffling independently for each replicate. So after the shuffling, we will end up having this. So these are the repeatability just under random expectation in this case. So let's look at my result for my first definition of repeatability. Here I use Dice coefficient, which is the equation given here, to quantify the overlap of DEGs of all pairwise combination of replicated populations in each of these 22 experiments. So the formula here is basically means, so for x1 is the number of DEGs in one replicate. X2 is the number of DEGs in the other replicate. So basically this formula is saying on average, what fraction of DEG in a replicate is also a DEG in the other replicate. So across all these 22 experiments, I found the median of the mean Dice coefficient is around 0.5. And they all exceed the random expectation, which is given by the gray dots here, typically by 25 to 50 standard deviations, as can be indicated by the z-score over here. And for the second definition of evolutionary repeatability, I obtained qualitatively similar results. So the sharing of DEGs with the same direction is also significantly greater than the random expectation. But here you might notice that the median of this mean Dice coefficient is, oh, sorry, I forgot to compute, but it's lower, definitely, by eyeballing, it is lower than 0.5, because this makes sense because we applied a more stringent criteria to calling repeatability here. But qualitatively, it is similar to the result that we just observed, which is the repeatability significantly exceeds the random expectation. For the third definition, the sharing of DEG with the same direction and magnitude is also significantly greater than the random expectation, even for the most stringent criteria applied. So here I define same magnitude as log2 expression ratio. So evolved divided by the ancestral is less than or equal to 0.5. So previously, because the previous example involves in experimental evolution, which we assume environment-specific selection, but the observed repeatability may also be explained by mutation bias to some extent. So I additionally collected an E. coli mutation accumulation dataset where natural selection is minimized. So and I repeated this, applied all three definitions of evolutionary repeatability for this MA dataset, because there could be a mutation bias such that makes a gene more likely to change. That is the first definition. A mutation bias can also make a gene to increase or decrease its expression. That is our second definition. For our third definition, there also could be a mutation bias that causes a gene to change by a certain amount here. So that's why I applied this to all my three definitions. And we can observe that for all pairwise combinations, there's still some repeatability observed when the natural selection is minimized. But the mean Dice coefficient here is generally low here. So I concluded that Some repeatability can be explained by mutation bias, but the contribution is minor, given the low Stiles coefficient observed here. Because I just observed that gene expression evolution is not 100% repeatable, I wonder whether different genes show different levels of repeatability. I focus on this E. coli long-term evolution dataset because of its large number of replicate populations. Here, I define repeatability as the number of is measured by the number of replicates in which a gene is a DEG here. If all genes have equal repeatability, the number of replicates in which a gene is a DEG should approximately follow a Poisson distribution, whose variance equals to the mean. And here, I observe that the variance of the observed distribution of the number of replicates in which gene is a DEG is much greater than that of the Poisson expectation, with overrepresentations of genes that show repeatability in 0 and at least 4 replicates. I finally investigated what kind of genes are more likely to exhibit repeatability. So here I use the same definition of repeatability as what in the last slide, which is the number of replicates where a gene is a DEG. I again use this E. coli long-term evolution data set. I hypothesized that genes controlled by more transcription factors are more likely to exhibit evolutionary repeatability. The rationale is that the mutation altering target is large if a gene is controlled by a lot of TS. In plain words, you are more likely to be hit by mutation if there's a lot of transcription factors controlling you. So I observed a significant positive correlation between the number of TFs controlling a gene and its repeatability in gene expression evolution. But the correlation, as you can see here, is relatively small, suggesting that the number of TFs controlling a gene is one, but potentially many factors influencing the expression evolution repeatability of a gene. So here are my takeaway messages. The number of gene expression traits exhibiting concordant changes in direction and magnitude between two replicate populations significantly exceeds the chance expectation for all three definitions of evolutionary repeatabilities. Genes controlled by more transcriptome factors tend to show more repeatable expression evolution, likely due to a higher certainty in the occurrence of mutation altering their expressions. And finally, in conclusion, phenotypic evolution during environmental adaptation is quite repeatable and deterministic, at least for gene expression traits. I want to give a few acknowledgements at the end of my presentation. I particularly want to thank my PI, Dr. Zhang, for his invaluable support. I also want to thank Drs. Yang Li, Angeli Mahyuka, Siliang Song, and Rui Qiyuan for their valuable comments on the manuscript. Finally, I want to thank all members in Zhang Lab for their support and kindness, and sometimes the random treats throughout the semester. And I want to thank you all for attention and thank you for having me. Thank you.
Thank you, Jiachen. I think due to time constraint, we can take only 2 questions. Do people have questions?
Yeah, I'm curious about the bigger data set with the multiple types of organisms. Did you see different levels of repeatability in different groups?
Yes. So if I go back to this slide, oh, can you see it? So this is my first definition of repeatability, which is the overlap of the number of DEGs between replicates. And you can see that the most repeatable one is actually this data set, yeast that is evolved in hydrogen peroxide. So here I ranked the repeatability from top, from it's more repeatable to least repeatable here. So indeed, there's some organisms that are more likely to exhibit repeatability, especially in yeast, mostly in E. coli here. For the like higher level, for like guppy or flies, they are not that repeatable relative to E. coli evolving in these environments, but still the repeatability exceeds the random expectation in all cases. Yep. Thank you for the question.
Thank you for the talk. It's very interesting.
Thank you.
My curiosity, I just want to know what you think about. So in my head, I'm thinking, while the differently expressed genes are very, very interesting, what are your thoughts on how they may be related to different phenotypes? Because could it be possible that while the genes may be expressed in a similar fashion, the result of whatever is happening may be different? So like a gene may be expressed similarly, but the phenotype in some interaction may end up being different, so that your end result is different while your kind of basis of that gene expression is the same. Or do you have any like thoughts on that?
I think that what you said also apply to other like organismal phenotypic trait as well.
Yeah,
Yeah.
just how the
Yeah.
reaction or the interaction of these differently expressed genes, and then maybe what the phenotype on a larger scale might look like. Even if your one gene is expressed the same in two pairs, but it may look differently because of different interactions going on between these genes, if that makes any sense.
Yeah, and I see what you're saying. I also consider that, and that's actually a caveat on my study. But in terms of if we consider the organismal trait, it is also hard to improve. It is also possible that the two phenotypes are interacting with each other as well.
Yeah.
So it is so hard to like isolate the effect from the any other factors. Yeah, this
Of course, I just
Yeah.
I just wanted your
Thank
thoughts.
you. This is very valuable.
Thank you.
Okay, thank you, Jiachen. Okay.
Thank you.